is production of proteins eventually based on the genomic sequence. And if you want to imagine the cell and the organism as like precisely a fine-tuned machine, you want everything to be clean, no unnecessary noise, and so on. But because of the complex nature of the gene expression, and also some of the genes are expressed at very low copy numbers, there is a noise that is often unavoidable. Okay? So then you like to understand how the noise arises and how the cells can actually utilize the noise even sometimes, or use other methods to suppress the noise. So this is a very famous paper published in 2002, and so obviously they are cells. What kind of cells? E. coli. That's true. And so most of the times when you see cells that look like that, they are E. coli cells. But there are some exceptions. There are some yeast cells, fission yeast. They actually look kind of like that too, although they are not as widely studied in biology as the case of E. coli. You can see that some cells are green and some cells are kind of red, and some others are yellow. So you see actually many different colors, and that actually is coming from the noise in gene expression, as I'll discuss later. So this is a paper that I wanted to discuss by Michael Elovich, titled Stochastic Gene Expression in a Single Cell. Thank you. Yeah, thank you. So there is a noise that you can call phenotypic noise. And you heard a term called phenotype. So you can have the same set of genes, but you may have different outcome, different phenotypes. So two isogenic cells, meaning that two cells that have the same genes, no differences in DNA sequence and so on, can have different numbers of identical proteins at a given time. So that is one example of a phenotypic noise. Although you have the same genetic background, the outcome can be different. The outcome in this case would be, some proteins have different copy numbers, between different cells. But where does that noise come from? If the noise comes from the fact that you have a small number of proteins, then this is called the intrinsic noise. And that's because when you have a small number, then there's always a randomness associated with this. So it was raining earlier this morning. So if you bring out a coffee mug outside and then collect the raindrops for 1 minute, okay, then let's say you collect on average 60 raindrops, okay? But then if you do the same next minute, you're not going to get 60. You may get 65 or 55, depending on, it's a randomness associated with this, right? So when your average is, let's say, lambda, so you have lambda drops on average per 1 minute, but sometimes you get, 59, 58, and so on. So you get variations. So that, how many, so if you plot number of raindrops you collect and the probability distribution, then it'll actually look something like this. And that distribution is called Poisson distribution. And the average value is lambda is the average. And that this function is look like this. that's a Poissonian distribution. So although your average is 60, then you have a broad distribution. Okay? So in Poissonian distribution, average value is lambda. Variance is also lambda. So you can show this mathematically. The variance is equal to the mean, but standard deviation, sigma is square root of variance. So it'll be the square root over n. So what it means is that if you were to measure the sigma here, that actually is equal to square root of lambda. So when lambda is 100, it'll be 10. So if the average is 100, then your actual measurement will give you 100 plus minus 10 of a single standard deviation. So this type of noise is also called shot noise. So if you want to, so noise in this case relative to the average value. So when lambda becomes large for large numbers, then the relative noise becomes smaller and smaller. Okay, so that's the idea. So when you have a protein expressed at a very low level, let's say 10 proteins in a cell, then the noise associated with this will follow Poisson distribution, and then you'll have a large noise about standard deviation will be a square root of 10 or 3, like 10 plus minus 3. That's a fairly large noise. Okay? So the intrinsic noise can occur because of the stochastic nature of collecting your raindrops using a mug when the average number of drops is actually quite low. And then all other sources of noise, for example, in overall transcription and transcription activities, can be dumped into one term. Okay, you have also other things like mRNA and protein degradation rates and local environments and so on. You can call them extrinsic noise. Okay. So cell A and cell B may have different numbers of the ribosomes. So cell A has more ribosomes than cell B. Then just because of that, cell A will express more of the proteins that you're studying compared to cell B, right? But it has nothing to do with the gene that you're looking at. So all these sources of noise are considered to be extrinsic noise.
What is the reason for the first noise?
Here? Actually, phenotypic noise will be phenotypic noise will actually include both of them. Okay. So what I mean by phenotypic is that two cells are identical genetically, but they have different outcomes, different copies of the same protein. Right? But when the copy number is small, then this can mainly arise from the intrinsic noise, shot noise that I discussed, Poisson distribution. When the copy number is large, then the extrinsic noise can be the main source of the phenotypic noise. Yeah. It's a little confusing. Okay. Sorry about that. So here's actually a very well-known figure that I took from a review paper. So fingerprints, and they are from twins. So twins share the same genetic information, but when fingerprints are formed, there is enough noise in the process so that even with the same genetic information, you get different patterns. Okay, so that's one example of phenotypic noise. Here's another example of a phenotypic noise. The two cats have the same DNA, but they have different fur patterns. So in fact, okay, this is called Rainbow, and this is the first ever cloned cat. Name is Rainbow. And when you do animal cloning, you get the DNA from an adult animal and put it into the egg of another animal, and then then you make a cloned cat. But the genetic material came from this one, genetic matter. But then, although the DNA is identical, they look quite different in terms of color of the fur and so on, and pattern. So that's another example of the phenotypic noise. All right, so I told you that if different cells all right, sorry about that. So now, if the exchanging noise dominates, let's imagine that you have two identical proteins you are making inside the cell, same cell, controlled by the same identical promoters, but you simply fuse them to green fluorescence protein and red fluorescence protein so that you can actually measure the fluorescence signal from a single cell as a function of time. Then when the exchanging noise dominates, let's say ribosome number goes up, then production rate for the both proteins will go up, and the ribosome number goes down, they both go down, and so on. So by following a single cell in two colors, you can deduce that it must be the noise is coming from the exchanging noise because they are going up and down together. Okay? And if you also look at many cells simultaneously, one cell will be brighter than the other, but if you look at the ratio between green and red, they'll all be the same. So everything will appear yellow. When you add red and green in RGB color panel, you get yellow color. One cell may be brighter than the other, but they'll all appear yellow. But let's imagine that the noise is now mostly coming from the intrinsic noise, so that these two different genes will actually fluctuate independent of each other because it's just coming from the same noisy process. So you can imagine doing it this way. So you have two mugs outside and then collecting the raindrops, and then if you're mainly collecting a lot, if you're on average you're collecting more than 500 drops every time, then the intrinsic noise will be very small. So whatever fluctuation that you see will be the change in the raindrop rate, right? So then they will go up and down together. But when you go down to very low rates, let's say 5 drops per minute and so on, then then each noise will be, they will experience noise just coming from the fact that they have a small number of events, and they will not be correlated with each other. So they'll just go up and down independent of each other. Okay, and if you sample single cells at a given time, then they'll have widely different colors. So if you, for example, catch this cell at this time point, then it will appear red. If you catch that at this time point, it will appear green, and so on. Okay, so by looking at a population of the cells and measuring their relative intensities of green and red, you'll be able to tell whether you get mainly exchanging noise or intrinsic noise. Questions? So in fact, this actually is the basis for this cover illustration that I showed you earlier. So the idea is that when you see a picture like this, you can say that intrinsic noise becomes very important in this experiment. So how did they do the measurement? They have E. coli genome, which is circular. Okay, in human cells, the DNA is linear. You have two distinct ends of every chromosome, but in bacteria, DNA is actually circular. So when DNA replication starts, it starts from here, called ODC. That's the origin of replication. And then DNA polymerases actually copy DNA moving in both directions. You have one DNA polymerase, one set of polymerase is moving in this direction, the other set moving in this direction. So DNA replication occurs in a bidirectional manner. And then termination of the replication happens somewhere here. So what they did was they replaced original gene in E. coli that is not essential with fluorescence protein of a certain color that I use green to denote. And then they also replace a gene at this location by another fluorescence protein of another color that I denote as red here. And so they are under the control of the lock repressor, same promoter, same everything. So they must be really essentially identically expressed inside the cell, and everything is the same except for the color of the protein. And then basically, when you look at the level of the two different proteins, they go up and down together. Then you can say that it's mainly extrinsic noise that is dominating, and vice versa. They had to do one additional trick, or they had to pay attention to the location of these two genes, because in a rapidly growing E. coli cell, DNA is copied actually even before DNA. You can start DNA replication even before the original replication has finished. So you have you can have many copies of the DNA. So if you have one gene somewhere here near the replication and another somewhere far away from the replication start site, then you may have more copies of this guy compared to that, and that can give rise to additional noise. That's why they made sure that they are about the same distance away from the original replication. Here, 1.5 megabase pace away, and then 1.46 megabase pace away. Okay, so the idea is that any difference between the expression levels of the two in the same cell may be called the intrinsic noise. Okay, first experiment. They use the E. coli cells. You can call them strains because they have it's a strain that has a certain genetic background. Minus LOCK-I. The LOCK-I is lock repressor. So this E. coli cell does not have a lock repressor in the genome. This means that if you have a lock promoter there, then there will be no repressor at all. So these two fluorescence proteins will be expressed without any hindrance at very high levels. Okay? So you then, when you look at the cells, they are very bright, and they are mostly yellow because both green and red proteins are actually highly expressed. So when you have green and red combined, you get yellow. Okay? So in this case, if there's any difference between the cells, that's not due to the intrinsic noise. It will be due to copy number differences of the ribosomes, RNA polymerases, and so on. Okay. The next experiment was to use LOCK-I plus strain, meaning that now you have actually the E. coli that has a lock repressor in the genome. Then overall expression level goes down for both proteins. So you have some repression. And so you make a few of these fluorescence proteins. So they become dimmer. But now you start to see cells that have mainly red fluorescence and mainly green fluorescence, in addition to the cells having a yellow color. Okay? So in this case now, you begin to see evidence of intrinsic noise. Questions? Yes?
Looks to me like they're more green than orange.
Yeah. Yeah. That I have to say that you can kind of play with the color scale when you present the data, right? So if the average of the two kind of matches, then you don't need to do any playing around. But if the one is expressed at a higher level than the other in general, then you can probably adjust the contrast to this. So, yeah, but that's a good point. All right. So, and then they added to this strain IPTG. IPTG is lactose analog. It'll bind to the lock repressor, so the lock repressor can come up from the promoter, and then no repression. And then even when lock repressor is inside the cell, these two proteins will be expressed at high level without any repression. So they both light up in green and red, and then they then in combination they appear yellow again. Okay? So that is the idea. Okay? Questions? Okay, so here I explain everything. What is B here? Huh? It's a panel B of the figure. Okay. How about RP22?
Would that be the strain of the bacteria?
Exactly. Yeah, it's the name of the strain that they used, and usually the people just give their own name. So if my students make a strain, then I would just call it TH25. Okay, Textipasle, whatever. So I don't know where but basically this is what you use if you want to contact the authors, to ask for that bacterial strain for your own research. So Neil, how do you name your strains in your lab?
So I made a plasmid that goes like P and K.
Uh-huh.
But I've never
NK is Neil Kim.
Yeah.
Okay. And P for plasmid, right?
Yeah.
Questions? All right, so then you can quantify the fluorescence intensity of every cell, intensity of the one color, and intensity of the other color. You see some variations. This is a linear scale, so if this is the average, and it can go down to 70% of the average, it can go up to 1.4 times the average in both directions. So, and you have data for 2 different strains, okay? And so which one shows a tighter distribution? M22, okay? And M22 is this one, okay? Without the lock repressor, you have proteins expressed at high levels. So relative noise is actually quite small, okay? And then D22 is the one that you have now lock repressor being expressed, okay? So now relative noise becomes larger. But if you have mainly extrinsic noise, then the noise will occur in these directions because both of them will be brighter or weaker, okay? But if you have intrinsic noise, that they will occur mainly in this direction. So you can see that actually when you go from here to here, extended noise increases in both directions, suggesting that you have contributions from both intrinsic and extrinsic noise. Questions? So if you pick a cell, this is a single cell there. It's strong in green color and weak in the red color. So that would be, oops. Okay, so if you go to an image like this will be more like that cell, right? This will be more like that one there. Okay. The really important point about doing this measurement is that, when you see this kind of scatter, how much is the real biological noise that is in the system, or how much is your measurement noise? Because measurement will always entail some noise, and then just looking at this alone, really, you cannot tell from just looking at the chart. But when you look at the image, then you know that this difference here, it cannot be just measurement noise. This must be real. So anyway, if you want to quantify the noise, then you really have to understand the noise sources of your measurement itself. Okay? Anything beyond that can be considered significant, but anything smaller than can be ignored. So they can also start with this strain that has lack of repressor being expressed, and then they can increase the IPTG concentration. So when you increase IPT concentration, the expression level of the proteins will go up. What happens is that then the intrinsic noise, which it was high at the beginning, will actually go down because now you have lots of copies of the proteins. So the shot noise will become smaller and smaller. And initially, the extrinsic noise actually goes up, and later on also goes down. So why does it go up and then go down? That is one of the homework problems for you to work on. Okay? This is very easy to understand, but why? Anyway, what you can say here is that at least at low IPTG concentration, low expression levels, that intrinsic noise actually can be much larger than the extrinsic noise. Okay? And by the way, this blue one is actually the total noise, as defined as square root of the sum of the squares of these separate noises. Questions? So this is a very important paper, and probably the most widely read and cited paper from this person. But so, what are the implications of the substantial noise measured here? Okay, they show that there is actually a lot of noise. But what does that mean? I just want to show a paragraph from the paper. So given the substantial noise measured here, reliable functioning of the cell may require genetic networks that suppress the noise, because actually the noise they're measuring was actually noise of an artificial system added to the native environment. So maybe in the native environments, there are some methods to suppress the noise of fluctuations, or are robust to fluctuations. Fluctuation may be always there, may always be there, but maybe cells have a way to cope with the fluctuation. And noise, however, if amplified, offers the opportunity to generate a long-term heterogeneity in a clonal population. And clonal population means that they are, again, coming from the same genetic background. You start with a single cell, and then it can double and quadruple and so on to form a population. But some cells can have very different properties from other cells, and that long-lasting heterogeneity actually may come from initial noise that is amplified. And that can be important for the survival of the bacteria as a whole, because when some cells are not really very active in, making proteins dormant. And then if you try to kill microbes by adding antibiotics that can kill the ribosome activities, maybe some cells that are active will be killed, but then cells that are dormant may not be killed and they may survive to preserve the genetic information. And then when things get better, then they can reproduce again. So this type of long-term heterogeneity may be actually useful for the survival of the bacterium as a species. Yes?
Can you show the graph of noise versus the fluorescence?
This one?
Yeah.
Hmm?
Why do you think the external noise is increasing? Why do you think it goes up? Yeah.
That is your homework problem. Yes.
The slide you just showed indicates the idea of long-term heterogeneity. So long-term scale of seconds, minutes, years. These things don't live very long, obviously, but
Probably more like an hours, yeah.
yeah.
Or days, yeah.
The other question that I guess goes along with that one, how long again were these bacteria observed? Was it possible to watch for long enough and observe individuals change color, or?
So let me tell you one thing. So two things. First of all, they didn't observe it as a function of time, and second, the reason is mainly because toxicity. When you shine light strong enough to see fluorescent signals here, then cells actually feel the light. Okay, so when you do time-dependent measurements, you tend to look at them very infrequently. Okay, so toxicity is an issue.
And of course, you wouldn't be able to keep track of individuals if you're just, flashing a light once in a while. Okay. Thank you. All right. So I want to mention one more study before I move on to the next topic. So 2 years after that paper by Alovich, Erin OShea, I think who was at San Francisco back then, now she's at Harvard. Then they published a paper doing the same experiment on eukaryotic gene expression. Okay, we are eukaryotes, but they didn't look at human cells, they looked at yeast. Single-celled eukaryotic system. And basically, their conclusion was that, well, maybe whatever Alovich said was specific to the bacterial system, because when they looked at yeast, and then look at two different genes, under the same control, the same promoter, but labeled differently in colors, they go up and down together. I remind you in the figure before, this direction was extrinsic noise, and this was intrinsic noise. Basically, there was not much to speak of in this direction, other than the measurement noise. So.
What are the colors?
These are different colors from different experiments. So this looks like 60 minutes, 90 minutes, and so on, different time points of the study.
Why do you have different colors in the left figure?
You got me there. So here, I don't know the answer to that question. Here you have different time points, 60 minutes and so on. The early time points, red are here. If you go to 36 minutes, 6-hour time point, they are all up here, right? So basically, they turn something on, and then proteins are being expressed from both gene loci, but they are kind of turning on at the same time, right? So it's possible that
that these guys actually had different histories, even in the same population, because sometimes in yeast studies, you can start a clock when the cell first divides. Okay, so cell can, and then, so that is a possibility. So I don't know, yeah, but this color difference may still be real. Yeah. But I think the claim there is basically that is small compared to the noise in this direction. So they calculate the intrinsic noise as a function of the mean intensity, and then extrinsic noise, and then one is much greater than the other. And then there were actually some other studies saying that actually it was true here, but it was not really generally true for eukaryotic systems. It's system specific and problem specific. But these two are really pioneering studies to quantify extrinsic versus intrinsic noise at the single cell level. Questions?
Oh, I realized that the intrinsic noise dips down.
I don't have an answer for that.
It is a revolution.
Hmm?
For yeast, the intrinsic noise dominates. It is a revolution.
Extrinsic noise down.
Yeah.
Yeah. Is this what?
It is more beneficial for them to do.
I don't think they actually know this at least in this case.
Or if you suppress the expression enough, can you still observe the intrinsic noise?
Okay, if you go to very, very low expression level here, right? But apparently even there, right? So one possibility is that they never really went down to really, really low level expression, right? So if the average copy number of the protein is 100 already here, right? Then that's not going to be, that your noise is just 10%, right? When your copy number is 100, and square root of 100 is 10, so 10% noise is already very small.
That
Right?
means the yeast is more productive in expressions than.
Yes. So I think what it means is that actually copy number has to be really, really small for the intrinsic noise to become significant. Yeah. All right, so that is a set of studies I wanted to discuss for noise and gene expression. I want to. I read a New York Times last evening, and I found this article that Fred Sanger died 2 days ago on Tuesday. Have you heard of his name, Sanger? Okay, some people are not. So, who is Sanger?
Oh, I've heard of his name. I don't remember who he is exactly.
Okay, so I knew what he did, but I didn't know that until I read this story that he won the Nobel Prize twice. And there are actually only 4 scientists who have done that. And one of them won the second prize as a Nobel Peace Prize, so he doesn't really count. Right? So who are the 4 scientists you know? Jean Bardeen, Marie Curie, him, Sanger, and Pauling. Yeah, Pauling. So Pauling got the chemistry prize and then the peace prize. I don't know what he did for peace. Maybe he smoked some weed. So Marie Curie won chemistry and physics. In fact, her family won 5 prizes. So pretty. Her husband got one, and then her daughter and her son-in-law got another. So basically, there are five prizes. Yes.
So how many people won 2 prizes in the same category?
One, John Bardeen, and there's 2. Yes, so he won the chemistry prize twice, and John Bardeen won the physics prize twice. Okay, so he won the prize that I knew about by inventing the modern DNA sequencing technology. In fact, his sequencing method was used to sequence our human genome. That was announced by, by like year 2001. Okay? So that method is called a Sanger sequencing method. But before, so in 1980 he won that sequencing prize, but already in 1958 he won another his first prize by developing a method to sequence proteins. Okay. So, and he actually somewhere in the article it said that he answered a question of which prize is more difficult to win, the first or second prize, and the answer is the first, because once you get the first prize, then you get a lot of resources to do more research. That you have to wonder why it's so difficult, why there are only 4. Right. But apparently he was not the type of person who would hire students or postdocs to the research. He did everything on his own. So he was the first author of the papers, and so even for the second one. And the excuse is that he was never good in finding projects for his people. So that's one way to win the Nobel Prize. So DNA sequencing. If you want to sequence the natural DNA found in long chromosomes, you first have to fragment the chromosomes into small pieces. And then these short fragments serve as the template. Then single-strand DNA that you want to sequence are primed with a short complementary primer. So that is a single-strand DNA you collect from the genomic source, and then you put short pieces of DNA called primer here. And then the DNA polymerase can bind and then extend this primer following this template. Okay? And then you do this by adding DACG to the reaction, and then based on the sequence, you copy that molecule. But trick that Sanger developed is this. He basically used chemistry to make what you can call halting nucleotides that can be incorporated, but that cannot be extended. I'll show this more in detail later. So then basically what happens is that then each replication continues until by some chance, then this kind of unusual nucleotide is incorporated, and then it stops there. Then you do a gel electrophoresis. You run it on a gel, and to see the length of the DNA, and then from that you can read the sequence of the DNA. So just to give you some more details, that is your template DNA that you want to sequence, and then you make a primer. And at the end of the this 3' end, if I recall what I discussed earlier about like the DNA nucleotide structure, you have this sugar ring. There's a 1' location to which the base is attached. 2' location, you have a hydrogen group. for DNA, and for RNA you have a hydroxyl OH group here, 2' OH, but for DNA is here. And here you have actually OH group as a function for the DNA. And usually, so you have an OH group there, so that the 3' end has an OH, and to which next DNA base can be added. But you can actually make chemically same molecule, but here instead of having OH, you have an H group. Okay, so this is deoxy. That gives you deoxy, but here you have another deoxy. Right? That's why this nucleotide is called dideoxy, DDATP. So if you mix DDATP with, other guys, and then by some chance, instead of ATP, this guy can be incorporated, then that once that happens, then DNA synthesis stops right there. Then you get a length, DNA of this length. And if it happens here, then you get a DNA of this length, and so on. But then you know that then actually, at this position, there must be an A. Right? So that is the sequence information you can get. By running it on a gel, you can measure the length, and you know that at that position, there must be an A. And then here, at these 3 positions, there must be an A. And then you actually you do this for A, and you do this for G, and C, and T. So you do 4 different reactions, and then you run it on the gel. And then so when you do this with the A, then you see all these bands, because actually dideoxy ATP was incorporated at that location. So that position must be an A. So that's like and so on. Then you read the sequence based on that. Okay, 4 lanes and 4 different experiments, and that's how you read the sequence. So actually, when my wife was a grad student at Berkeley 20 years ago, she used to run sequencing gels like this. And so and then you can do maybe 150. You start from 1 here, 80, and then 140. But it's very laborious. Takes a long time.
So when they run down the gel, are they in one template DNA and then one that has stopped along the way?
That's right. So basically, this is one template DNA, but with a DDATP. Okay, and then you can stop at different positions, right? You have many actually products of the reaction.
So they're not, when they are running down the gel, they're not single strand. They're not fully double strand.
They are single strand.
They are.
They denature the DNA, and they don't only look at the top strand, newly made one.
So they put the same thing in 4 different lanes from the same.
Yeah. But then they did the reactions with 4 different DD nucleotides. So here you do the reaction with the DD-GTP. Then it stops at G locations of every G, right? And locations of every C, and so on. And here actually what they did was you can see the right 4 lanes loaded 90 minutes after the left 4 lanes. What it means is that so there was a time difference of 90 minutes between the two. It's because DNA molecules, if you apply electric field, they migrate from here downward. For the shorter ones migrate faster. For this one that had extra 90 minutes, the shorter ones already have migrated down the gel. You cannot see them anymore. You see the longer ones only. And because these guys are loaded 90 minutes later, then you can see the shorter ones here. So by running one gel with 8 lanes, you can read 140 base pairs. Okay, but it can be very laborious. Okay, so this was still commonly done 20 years ago. But in 1987, some visionaries actually got together and they said, we're going to sequence the human genome, 3 billion base pairs. When a student had to run this gel to get 140 base pairs in one afternoon, right? It's a pretty crazy idea. But it got better because they then resorted to fluorescence-based detection instead of running a gel. But the idea is the same Sanger sequencing, but you read it by using fluorescence. So that made the sequencing faster and cheaper. So basically, that is still what was used to. Sorry, this is the wrong slide I have. The sequence of human genome, and that's the chromosome 1 has 279 million base pairs, 2 has 251 million base pairs. If you zoom in 5,000-fold, you see a gene for beta globin. This is actually a protein that makes up hemoglobin that carries oxygen in your blood. And then if you zoom in further, then you see the DNA sequence, and then further, then finally you get down to the level of individual bases. So the sequencing was completed in 2001 or 2, and the insights gained from the human genome sequencing, well, you have about 3 billion letters, A, C, T, G. An average gene, for protein, is made of about 3,000 bases, so about 1,000 amino acids, because one amino acid requires 3 bases. And the largest known human gene is more than 1 million bases long. And the big surprise when the result was announced was that among the human genome, the protein coding genes were only about 30,000 different estimated genes instead of many more. So we do not have any more genes than other animals or even very primitive organisms. So that was a big surprise. And other than protein coding genes, we also contain lots of genes that code for RNA that are not coding for the proteins, so and so on. And almost 99% of the nucleotides are the same in all people. So back then, functions of yes.
I was wondering how, just looking at the sequence, you can tell where a gene is and where it is not a gene.
So that is a good point. So the main thing is done by looking at the codons. So for the translation. So when ribosome translates proteins, it has to start from a certain codon, a start codon and a stop codon. So basically, I think they run a search, and then you find a stop and start codon separated by a certain distance. Okay. So, and then probably there are some other criteria that was used.
You show that the size of genes vary a lot.
Mm-hmm.
You know what the distribution
Whereas the average is apparently 3,000, right? But I don't know this distribution.
And in a single experiment, you can sequence like 200 base pairs.
By running a gel, yeah.
Yeah, then how do you connect the different results to get the whole gene?
Yeah, that's a very good point. So you do. So nowadays, people use what's called a shotgun sequencing. You just sequence lots of small fragments, and then you align them, put together into a single DNA using a computational algorithm. So that was the approach proposed by Craig Venter, who is a very flamboyant, person who may win the big prize. And so, if I make an analogy, I think once I had, I guess I still do the same, but Netflix doesn't quite work. But I had ordered a cable TV service for my home, but then the company made a mistake, so they didn't actually block certain things that I didn't order, right? So I had, Showtime, HBO, everything, So what I did was that whenever I had like 10 minutes, I just sat and then just watched like TV for 10 minutes, and then it's like sequencing 100 base pairs of DNA, right? But if you watch enough of those pieces of Crouching Tiger and Hidden Dragon, whatever, then you can actually reconstruct the entire movie, right? So that's what I did, and then that's what they were able to do. And this is difficult for certain types of sequences, when you have many repeats. Okay, so less than 2% of the genome codes for proteins, and repeated sequences that code for proteins or junk DNA, that's at least half of the human genome. And you see repeated sequences, people didn't think there is any actually function for them, but maybe give you some information about the genome evolution. And so on. So that was the human genome project, and they use a Sanger sequencing method. So I think he certainly deserves the Nobel Prize. So I'm plotting output per instrument run. So you have a commercial DNA sequencing machine, and you run it once. It takes maybe half a day or whatever. Then how many base pairs can you read based on a single run? Output in kilobase pair. And this is as a function of time. Okay? So when this human genome was announced, the draft human genome was announced in 2001. Back then, the single run of an instrument gave about 100, up to 100,000 base pairs. Right? There's 1,000 base pairs and 100 here. And then it stayed at the level for like 3 more years. And then it jumped here, and jumped here. And then now, 3 years ago, we were doing more than 10 to the 16 base pairs per run. Well, it's more than that. It's 10 to the 14. So that's not Moore's law, it's a hyper or Weber Moore's law, right? So something actually happened somewhere here that really allowed the sequencing machines to be much more rapid. Okay? So, nowadays basically we don't use Sanger sequencing to do DNA sequencing anymore. At least at the large-scale sequencing projects, Sanger sequencing method is not used anymore. Okay? So what they use is called the next generation sequencing technology. There are several of them, but you can see from this chart, nowadays Illumina is a company that dominates the sequencing market. And the company, last time I checked last week, the market value is $12 billion based on just that sequencing technology products. Okay? And so what happened was that Illumina's actually predecessor, so-called Solexa, actually product debuted right here. So that's when there was the major jump. Okay. And then after that, actually, there are some other competing technologies, but then this guy just continue to improve and still dominates the sequencing market. In fact, it is actually so dominant, and things are moving up so quickly that you can buy a sequencing machine from Illumina from like 3 years ago from eBay for like $3,000, whatever, because no one in the right mind would actually use the machine from 3 years ago, because if you use that, then sequencing cost will be like 20 times higher, right? So then you have to upgrade. Otherwise, it doesn't make sense to use yesterday's technology because today is so much cheaper. Questions?
What's the advantage to continue to push the output of these machines if you're beyond the length of any genome that you're going to look at? I don't need 10 to the 17 base pairs in a day because I only am interested in a billion, right?
Yes, but that also means that it's getting cheaper and cheaper, right? So now I think these days human genome sequencing costs maybe $2,000, and the initial draft this cost $2 billion. Okay? Now if it goes down to $200, then when a cancer patient comes to the hospital, at the first thing that you do, you sequence, tumor at different positions, and then you sequence a normal cell, and then that'll tell you exactly what's going on at the gene level, and then, insurance companies can pay for that, right? But not when it's, $10,000 or $2,000 and so on, because if it's cheaper, then instead of just sequencing one piece of tumor, you can sequence 10 different pieces, or as a function of time of treatment and so on. Nowadays, people use sequencing to test for Down syndrome. Down syndrome is actually due to the extra copy of a chromosome, and in the past, the test that you could do to get a definite read of the Down syndrome extra chromosome was to do some invasive procedure to insert a needle to collect some materials from the fetus, right? But that has actually its own problems. But nowadays, what they do is that they just use sequencing of the mother's blood, because blood contains DNA from dead cells of your body. But then if the fetus is there, then there's also DNA from fetus cells. And of course, fetus gives you a very small amount of DNA, but because sequencing is not very cheap, you can sequence just a lot until you have enough signal to the noise ratio to show that the fetus has 3 copies of the chromosome instead of 2, right? So there are 4 companies offering the service, and the price is $2,000. Or actually, the actual cost of running the test is probably $100, right? They're doing it at an enormous profit. And well, I think in California alone, like last year, 150,000 women took the test. And pretty soon, insurance companies will pay for it, because basically it's so much better than putting a needle into the fetus, right? That all of this was made possible by this jump.
How does fetus DNA go into the?
Well, cells, even in the developing animal, some cells are killed in a programmed way, programmed cell death. These cells will have to be destroyed, right? Then DNA is not fully chewed up and then goes into the circulation. So same kind of people also are using this test, this kind of test to look at the organ rejection. So somebody gets like heart transplant, whatever, and whether the organ is getting accepted well, usually they have to open it up and then to get some sample. But nowadays, you don't actually have to do that. You just look at the DNA in the blood, and if the organ is getting rejected, then cells will die much more quickly. You'll find actually foreign DNA there. Right. Again, it's really a matter of it's mathematics, and signal-to-noise ratio. And when you have a cheap DNA sequencing method, you can do it until you have enough signal-to-noise ratio. So I believe actually much of the biological research will be done by doing sequencing. Many of the laborious tools that people used before will be replaced by sequencing, and they are already being replaced. So I want to describe actually some of these applications next week. But next week, no, after the Thanksgiving. But today I want to discuss this Illumina sequencing method. So let's see. So this is how, why don't we just look at the movie first? Okay.
The Illumina Genome Analyzer sequencing technology. The key to the genome analyzer's unmatched flexibility, accuracy, and ease of use.
Can you hear?
Yeah, other keys.
Let's look at the chemistry.
There's a speaker here?
Steps in the genome
Ah.
analyzer workflow. Library preparation, cluster generation.
Library preparation. The Genome Analyzer performs massively parallel sequencing of hundreds of millions of fragments of DNA. First, the DNA is fragmented. Sheared ends are repaired and adenylated. Adapter oligos are ligated to both ends of the fragments. These fragments are then size selected and purified. Cluster generation. On the CBOT cluster system, single molecules are isothermally amplified in a flow cell to prepare them for high-throughput sequencing. The 8-channel genome analyzer flow cell has a dense lawn of oligos grafted to its surface. These oligos bind to the adapters ligated to the library fragments. Single DNA molecules hybridize to the lawn of oligos. Bound fragments are extended to create copies. These copies are covalently bound to the flow cell surface. Each library fragment is clonally amplified through a series of extensions and isothermal bridge amplifications, resulting in hundreds of millions of unique clusters. The reverse strands are cleaved and washed away. Ends are blocked, and a sequencing primer is hybridized to the DNA templates. After cluster generation, the libraries are ready for sequencing. Sequencing. On the genome analyzer, hundreds of millions of clusters are sequenced simultaneously. The DNA templates are sequenced base by base in parallel, using 4 fluorescently labeled, reversibly terminated nucleotides. All 4 bases compete with each other to bind to the template. This natural competition ensures the highest accuracy. After each round of synthesis, the clusters are excited by a laser, emitting a color that identifies the newly added base. The fluorescent label and blocking group are then removed, allowing for the addition of the next base. This proprietary chemistry reads a single base in each cycle, enabling accurate sequencing through difficult regions such as homopolymers and repetitive sequences.
Okay, so let me go through them a bit more slowly. Okay, so you get genomic DNA into some fragments, then you can connect what are called adapter DNA sequences to both ends. You can see different colors, and then you have a lawn of little small DNA molecules, and then what happens is that you can anneal that adapter sequence to that one. So then it stands up on the surface. And then you use a primer to make a copy of DNA. So this is called a bridge amplification. So you make 2 copies, and then you denature them. Then you have 2 copies of DNA standing up. And then you do additional bridge amplification. Then you get 4 copies, and then you get 8 copies, and so on. So basically, once one single DNA lands here, then this can be amplified into, thousands of copies of this identical DNA sequence in that area, forming a single cluster. Okay? So you do this copy, and you denature, and then you do it again and again. And then you get basically clusters of the identical DNA molecules. And this is another cluster. of a different sequence coming from different genomic DNA fragment. And then what you do is that you read the sequence off by using fluorescence. And you add 4 different nucleotides, A, T, G, C, but with labeled 4 different fluorescent dye molecules attached. And then you use laser fluorescence to look at which color this cluster has. Okay. The beauty of this is that it's not a single molecule method. You have thousands of DNA molecules. So if some of the reactions not work really well, or dyes are photobleached and so on, it doesn't matter because you just see a very excellent signal-to-noise ratio. The color is very clear, and then you do it many times to read the sequence. So the key parameter is the read length. Basically, how many bases can you read before your signal-to-noise ratio becomes too large? And they have really improved on it. So you can easily do 1,000 bases of DNA reading this way. Of course, when the read length is larger, it's much easier to do shotgun assembly, right? So if I watch Crouching Tiger and Hidden Dragon, is that right? Or Crouching Dragon or Hidden Tiger? Oh, anyway, if I watch it in 30 minutes increments, it's much easier to assemble the entire film, right? So that long read length actually is another improvement that people have made over the years. So that is the idea. And the beauty of this also is that it's not a real-time method. You add these, and then you just take an image, and you take an image of the entire microscope slide, 100 million clusters, right? So it's highly parallelized. So if you have 100 million clusters, and each one giving you 1,000 base pairs, that gives you how many? 100 billion sequences of read, right? So that's what they can do. So it's really amazing technology. I believe I mentioned this before, but this company, Solexa, was the original company that was purchased by another one to make Illumina. That company was started by 2 assistant professors in Cambridge, UK, in chemistry, and with the idea of doing single molecule sequencing. It's quite amazing, actually, how far, you can go, right? So, from 2 assistant professors to a $12 billion company. There's another competing technology, which has actually non-negligible market share. It's actually a company called Ion Torrent, and so in this case, you don't need to read anything here, but basically, you're not doing fluorescence measurements to read the sequence. You're doing electrical measurement. How do you do that? Well, when DNA is extended by 1 base by adding the nucleotides, then in the reaction, protons are released, and then you just read the protons using electrical measurement. You cannot read single proton using the device, but because you do bridge amplification to make lots of copies. So if you have 10,000 protons being released per reaction, then you can detect it easily electrically. And the beauty of this approach is that you can use mature semiconductor electronics industry fabrications, so you can make it highly parallel. Okay? So that is the one promise of that method. So basically, this is not a fluorescence that just colored just that way, but A is added, and then when A is incorporated, a proton is released, and then you will register a certain current, right? But if you add T's, and then there are 2 successive T's to be incorporated, then you have twice the protons level, twice many protons will be released. So your readout will be actually twice as big. So then what you record is, oh, now I have A and 2 T's, and so on. So that's how they do these type of measurements. Of course, there are problems. When you have like 10 T's, then 10 times the unit current is more difficult to distinguish from 9 times compared to 2 from 1, right? So when you have a large repeat, they also get into some trouble, and you get some errors there. Yes.
Here, do you go in one kind of nucleotides? So
That's right. Yeah. So what they do is that they just do A, and then wash everything away, and then put T, and so on. So this machine you can buy for $50,000, and you can put it in your office. It's the size of a laser printer, right? Because you don't need to have a camera or laser, right? And you can sequence the bacterial DNA for just $500. All right, so I want to also talk about the next generation DNA sequencing methods after the Thanksgiving. No, that's right. Yeah. Okay. Happy Thanksgiving.